So my name's Alex. I am sixth year at PhD in Cosgrove Lab. I work on looking at the signaling dynamics in the muscle stem cell niche in both a wet lab and dry lab sort of perspective, where I do things such as like interrogating phosphoprotein signals and how they get activated in response to diverse stimuli, looking at like linear regression modeling of those sorts of signals, and then also uh, like looking at methods for expanding muscle stem cells in a long-term culture environment. So I think I'll let someone else explain what they do, I guess.
Yeah, maybe I should go next. So I also work in the Cosgrove lab, but on top of that, I'm jointly advised by Ewan DeBlanek. And so my work is kind of surrounding genomics, which Ewan's lab is working a lot on, and muscle stem cells, which the Cosgrove lab works a lot on. So we're using a variety of genomics, transcriptomics, and bioinformatics strategies to explore the roles of non-coding RNAs and also niche factors and muscle stem cell expansion and differentiation and a couple other areas, things like that.
Do you want to go, Aria?
No, you go ahead.
Okay. So I'm a first-year PhD in the DeLisa lab, who's actually field faculty in the chemical and biomolecular engineering department. And my research so far has been focused on more of protein engineering with proteasomal systems inside, and kind of taking advantage of the facts in the E. coli to try to, and E. coli, yeast, and mammalian cells, in order to be able to actually degrade whatever target protein that we'd want, particularly, hopefully, cancer target.
All right, and that leads me. My name is Aria Henderson. I'm a first-year PhD student in Dr. Isaac Lee's lab. We are, in general, a vasculature on chip lab. Oh, hi, Toby. We're a vasculature on chip lab. My specific project uses a microfluidic device to look at the interactions between pericytes and lymphatic endothelial cells, specifically with a focus on the how that is affected in the inflammatory environment, with an application towards lymphedema. We also have some tissue engineering projects that we do in the lab as well, and some cancer-related vasculature on chip. But in general, we're working on in vitro models for vasculature.
So with that, I can talk a little, I can answer questions people might have. I can talk a little about what it's like working with my PI. I could talk about why I chose to study what I'm studying. There's lots of different things we can discuss, but it's really, I guess, student-driven in a sense.
Feel free to ask questions in the chat.
All right, so I would say my work is definitely a hybrid of computational versus experimental. So in some of my earlier studies where I was looking at sort of like the signaling pathways that regulates phenotype in muscle stem cells, I did a lot of the gathering my own data with a whole slew of 12-well plates that had to be timed with certain ways to measure phosphoprotein activation. And then I fed that into my own data model that would inform like for linear regression to see if we could predict phenotype based on signaling dynamics earlier in the process. So mine is definitely a mix of both. I think other people might be more dry lab. What do you think, David?
My work is also pretty much 50-50. So I kind of have two main projects. One of them is more methods development for developing single cell RNA sequencing strategies. And the other is completely dry lab. Like I said before, I work in the Cosgrove and DeBlanek labs. When I'm in the Cosgrove lab, I'm kind of I'm the computational biologist, and when I'm in the Diplomic lab, I'm the biologist, just because they're very different groups with different types of people in them. But yeah, I'd say I'm also about 50-50.
I pretty much only do experimental stuff. Yeah, I'm only the first year, so I might have to do computational at some point, but I don't really enjoy computational as much, so I'm kind of hoping and gearing my research towards just experimental stuff. But there are some people in my lab that are doing a lot of computational things, basically with just a ton of DNA stuff that they've been able to get, but I personally, I wouldn't want to do that, but there is lots of opportunity to kind of reach into both pots.
And from Lead Lab, well, first of all, I'll say that we're a brand new lab that just opened in July. So we've only been around for less than a year, but so far we don't have any computational biology or any computational projects in our lab. We're very much wet lab focused, with some like immunofluorescence to look at the different cell expression that we see in our models, but so far no computational work.
Maybe to just briefly chime in, I think there's probably relatively few labs that do exclusively computational. So I think most of the labs, I think in BME, there's quite a few that do both experimental work together with computational, but I think there's very few labs where you do exclusively computational. I think Haiwan Yu's lab, he's one of the graduate field faculty members. He has people who work more on it in terms of looking at protein-protein interactions, some of the systems biology there, but that's probably more the exception than the rule. So I think in most other labs is you generate data and then you can use computational models and all kinds of modeling for it.
So I guess maybe we can move on to the next question. Synthetic biology community. That's a good question. I can't think of any really like classical synthetic biology labs off the top of my head, but I mean, there's a lot of really cool work being done in like Alana Brito's lab and looking into kind of basic science questions in microbiome, in kind of the view of the microbiome and things like that. Anyone else have any good examples off the top of your head? Maybe Connor would know.
Yeah, from what I know, that there's a lot of so I don't know. I think synthetic biology is just such like a broad topic. Like you can include so much in that. I think a lot of if you are interested in more synthetic biology, maybe looking at a little more of the field faculty in some of the different departments, because I know there's some in the biological sciences, and then also like the department I'm in, the CBE department, has a lot of people doing a lot of different things with glycosylating proteins using synthetic biology and a lot of different other applications.
Yeah, I just added, I think, for example, Matt DeLisa, I think they work a lot on glycoproteins on the surface. So he's graduate field faculty. Chris Alabi, I think, is another person who would probably just come to mind as well. And so they're in the Department of Chemical and Biological Engineering, but they're graduate field faculty. We have students who are working in their labs as well. So I think those would be some of the names. Matt Pashek, he's done some other work as well.
Any other questions from the crowd? I feel I always get the question of how much coding experience I had coming into this within the kind of the world of computational and systems biology. And to answer that before anyone asks it, I mean, there's a whole wide range. Like I know Ewan's lab is about a third computational biologist, so I have probably the least amount of coding experience coming into that lab of all of the grad students. But it's a wide range. And a good thing to remember is that you're going to grad school. So you're expected to learn things when you get there. You're not expected to know everything when you start. but other questions are welcome.
Ooh, we have a mentoring style lab culture question. Who wants to go first on that one? Okay, I will. So Lee Lab, like I said, we're a very new lab. Our lab culture, I guess, is still being kind of formed this year, which is really fun. I like being involved in the lab formation process. But we did start doing experiments in October, so we got the lab functioning pretty quickly. Dr. Lee claims that his mentoring style is a little bit more hands-off, but he is a brand new faculty member, so we're still working a lot with maybe ideas he had as a postdoc or ideas that he's trying to set up now to eventually create his tenure package, his tenure portfolio. So he's still like very hands-on and active in the environment while we're doing training. He has offered, he said, if you ever want to receive any direct training from him, go ahead. And I did actually take him up on that when we were making our chips, and he ended up coming in on a weekend too, because it's a 6-day process, and so you have to come in on a weekend for that. So he's really open to suggestion. He's brand new faculty, so he is very excited with science and really likes all the different possibilities that you can take projects in the lab. So he's kind of flexible in that way. I will say that his mentality on projects that he always says is undergraduate students and postdocs can do risky projects because undergraduates don't need publications to graduate, and postdocs need really great publications to go get their professorship, whereas he prefers graduate students, like PhD students, to have a little bit safer project, at least to start out with, so that we're guaranteed to get some data and some publications so that we can graduate in a timely manner. I know with the project that I'm starting right now, this is kind of like my starting project because it's a little bit safer, and then I'm going to go into a little bit riskier project, probably in my 3rd or 4th year. And then lab culture, we're not a very early morning rising lab, so we're there in the afternoons and the evenings. Our devices require you to come in at least for an hour or 2 on the weekends, so we're there pretty much 7 days a week. But because of that, Dr. Lee is very flexible about what times we do come into lab, and that's why we don't have to come in the mornings. And we're a really fun lab. I don't know, it feels like family. It's cute. All right, I'll let someone else go.
So I guess I could talk a bit about the culture of Ben's lab. So I would say that Ben is a rather hands-on sort of PI. He likes knowing some of the day-to-day details. He's a type where he always basically has more or less an open door policy, where if he's there, you can talk about research or things going on in the lab. I would say that the lab culture is pretty relaxed and friendly to an extent. I mean, David could probably allude to it a little, because I had a bit of a different experience because Ben was just starting when I started. Now, David has been in our lab long enough, or like kind of a more mature lab in a sense, so where things are a bit more established. Whereas when I was there, it was maybe like 1 or 2 PhD students at a time. I really enjoyed being in our lab. We have fun together. It's good. But I know David has probably other similar but different opinions.
Yeah, I mean, I think most PIs in our department have an open door policy. I know Jan has an open door policy as the DGS for everyone. Yeah, that's, I think, pretty ubiquitous. But like Alex said, yeah, I've been in the Cosgrove lab just since it's kind of matured a little bit. Ben just graduated his first student. The DeVlamic lab is kind of on the same timeline. So Ewan also just graduated his first student this past fall. So it's been kind of nice to be in labs that have just kind of finished developing their own work cultures and things like that. Yeah, I mean, it's pretty lab to lab. Like, these cultures are specific lab to lab, so it's hard to answer for like the broad community. But I know a ton of labs do like dinner events and things like that. Like, Cosgrove Lab and William Lab both do lab dinners at least once a semester. Cosgrove Lab has a great Christmas party every year. Yeah.
And speaking for the DeLisa Lab, I mean, we have, so DeLisa's has been pretty established. So, I mean, just last year he graduated like 5 people, I think. So he just got a ton of people this year too, although he's still looking for more. So we have about, I think, including grad students, undergrads, and postdocs, we have about 20 or so people in the lab. So since there's so many of us, he tends to be a little bit more hands off. We still have like weekly meetings, and then but then we also have smaller groups that we meet every 3 weeks. And then after that, we basically just do our work without him, as long as we give him results, then he doesn't really interfere as much. So most of all the lab areas and everywhere where we're actually doing the research is pretty much maintained by everyone in the lab. So we have a kind of a good community and a lot of people to kind of trade off jobs to be able to really work together. But we still have we usually have like a monthly outing, whatever that may be, for the month. We usually vote on it, whether it's like a dinner or a lunch, or if we go out to like a bar or something, and which is paid for by the department or the lab. And then but so we're all pretty friendly, I think, and it's kind of nice to be able to have the community that has a lot of people, but then you still have like interactions with your professor, but you still have some level of autonomy that may not happen in some other labs. But it's just a personal preference of what kind of lab you'd want, and that's pretty much what the rotations are for.
I do want to add one little interesting tidbit about being in Lee Lab. Right now, our undergraduate students work individual projects, so they're not assigned to be with a specific grad student or postdoc. And because of that, it kind of has a very teamwork, co-collaborator feel at all levels, especially since all of us started in the lab this past fall. None of us have a strong experience in Lee Lab, so it's a very much nice, collaborative, even playing field sort of environment. And I also want to throw in the tidbit that all birthdays get celebrated in the lab too, so that's pretty cute. We have a cake, and all of our birthdays seem to kind of group up, so we celebrate in clumps. But my previous lab didn't celebrate undergrad birthdays because they didn't consider undergrads part of the lab. But here at Lee Lab, we consider undergrads full members.
Any other questions from the crowd? Anything about resources that we have in the department? Maybe I can mention BioHPC, our high performance computing group that everyone has access to. And they've got some pretty big clusters that we can work on. Different labs have their own computer clusters, so the computational facilities at Cornell are actually pretty impressive. I don't know, anyone else have anything to mention?
I mean, kind of in addition to the BioHPC, we also have the core facility, which has a lot of machines in different areas. So that's where we do all of our DNA sequencing, and also we use all the flow cytometers there, and then another thing for our professor just became sort of the current president of that sort of area of all the BioCore facility. So he's pretty busy, which is why I think also he's a little more hands-off. But there's a lot of resources for our research, and even if you don't have something in the lab, I know a lot of times if we don't have a machine, we can go to a different lab and just be like, hey, can we borrow this for a while? And there's a lot of cross collaboration between a lot of the different PIs.
And
yeah, Sorry, just to add to what Connor said, the flow cytometry core just got a ton of new machines. Jan might know the numbers better than me, but several $1 million and new sorters and everything. And also the sequencing core is quite good. We do a lot of single cell RNA sequencing here. I know we've started doing a lot of spatial RNA sequencing as well. So there's a lot of cool toys.
And then I was just going to mention that Cornell has a really rich imaging history and background. So we have a lot of different imaging facilities, both here and then some of the labs in New York as well, that we work with to get some really great images, working anywhere from like cellular all the way up to like organ systems.
I think that's you, Connor.
Yeah, that's just for you, Connor. The rest of us are BME labs.
Oh yeah. Well, I'd have to say, not really, because there's still a lot of like BME specific. Like we have the seminars roughly every Friday. We still have like the work in progress seminars that we go to. So I still and with our cohort, we also, like when we first came here, we started a Slack. So we've been basically slacking, and like we have all these different channels, and so I think that we've done a really good job of like kind of staying together, even if we are in a lot of different labs. Even because like some of the BME labs or part BME labs might not even be in the in WOW. So, I think there's still a good job of kind of keeping that community, even if you're not in the same lab or in the same building.
And I'd imagine some of it might depend on the labs as well. I think there's some labs that have quite a few BME students. So I guess Dave Erickson's lab, I think you have about four of the BME students probably at least in your lab. I think Matt Paycheck's lab, Matt DeLisa's lab, each of those labs probably has 2 or 3 BME students in it. There's other field faculty where there's maybe just a single BME student in there, but again, they usually then still find other ways to connect with the BME community. Looks like we've exhausted everybody's questions. So if there's no further question, I think we'll just conclude the meeting here, since I think we have the next session starting in 3 minutes. So thank you everybody for just taking the time. And just if there's any remaining questions, feel free just to reach out to these students. We can connect you by email to them as well. If you want to get a virtual tour of any of the labs, see what it looks like, I'm sure we can arrange something like that. Although I guess now with everything shut down, it might be a little bit harder to do, but we'll see.